The overall goal of this procedure is to demonstrate the application of FlexStation 3 to study the pharmacological profile of the transient receptor potential, or TRPA1, channel. This is accomplished by first preparing cells that express TRPA1 channels in 96-well microtiter plates. After washing the cells in a microplate washer, the cells are loaded with the calcium-sensitive fluorescent dye Fluo4AM. A cell loading period is then followed by several additional washes. The next step of the procedure is to prepare the compound plate, including a TRPA1 agonist and a putative TRPA1 antagonist. As a final step, the software parameters are set up for plate reading in the FlexStation 3 system. Ultimately, results can be obtained that show dose-response relationships for TRPA1 activation by flufenamic acid and inhibition by the antagonist compound A.
Though this method can provide insight into TRPA1 activation mechanisms by measuring calcium influx, it can also be applied to study functions of other calcium-permeable ion channels and G-protein-coupled receptors using calcium-sensitive fluorescent dyes or memory potential dyes.
The main advantage of this technique over existing methods like calcium imaging and manual patch clamping is that this time-saving technique provides high-throughput screening for the function and modulation of calcium-permeable ion channels and other calcium-mobilizing receptors.
Prior to the start of this protocol, grow HEC293 cells stably transfected with TRPA1 to a density of 70 to 90% as described in the written protocol. To begin cell preparation for the calcium assay, coat 96-well plates.